Day. Welcome you all for the course Research Methodology. Today we have got Unit 13, Testing of Significance. So in the previous session, we were talking to you about the framing of hypothesis. So I was just explaining you about how do you frame the null hypothesis and the alternate hypothesis. In this session, I will be going through how do you test an hypothesis, and I'll be showing you the test applied and how do you interpret with the data what you get. So here the alternate hypothesis, usually the initial hypothesis that's going to predict a relationship between two variables. But the null hypothesis is the prediction of no relationship between the variables that you are interested into. We wanted to test whether there is relationship between gender and height. That is the first example that we are taking. So based on the knowledge of human physiology, we formulate an hypothesis that men are on average taller than women. Okay. So to test this hypothesis, you restate it as H naught is null hypothesis is men are on average not taller than women. Okay. The alternate hypothesis is just the reverse of that. So men are on average taller than women. So the steps what we need to follow for hypothesis testing is we formulate two hypothetical statements H0 and H1 such that only one of them is true. By doing so, we can check the validity of our own hypothesis. So the next step is to formulate the statistical analysis which we are trying to follow based on the data points. Then we analyze the given data using the methodology. So the final step is to analyze the result and judge whether the null hypothesis will be rejected or we fail to reject the null hypothesis. That's what we do. So here, when we test a hypothesis, we need to look into the normality first of all. We need to first check whether the population is normal. So this type of testing is used for normal distribution. If the data points are grouped around the mean, the probability of them being above or below the mean is equally likely. So it resembles a inverted bell-shaped curve and it shows an equal distribution. So we have our mean and standard deviation, where the standard deviation should be lesser than the mean value, which means that the sample is normally distributed. So only for the normal distribution data, we are supposed to go in for testing of hypothesis. So that's the base thing what everyone should have in the mind. So first thing is the t-test. Normally t-test is done for samples which are lesser in size. We call it a small sample. Okay. So here if it is lesser than 30 only, we go in for that. So this test is used when it is normally distributed, the population is normally distributed, and standard deviation is actually unknown. And if the sample size drops below 30, we use t-test to find the confidence intervals of the population. Then comes chi-square test. This test is used to test the population variance among the known or assumed values of the population variance. It's also a better choice to test the goodness of it. Goodness of it indices is being here of a distribution of data. So the two most common chi-square tests are chi-square test of independence and chi-square test of variance. Then we have another powerful tool, which is ANOVA. The short form is ANOVA. It is analysis of variance. It compares the data sets of two different populations or samples. It is actually similar to t-test or the z-test, but it allows us to compare more than two sample means. So the ANOVA allows us to test the significance between an independent variable and a dependent variable, namely x and y respectively. So we have got z-test. It is a statistical measure to test the means of two population samples, whether they are different when their variance is known. For a z-test, the population is assumed to be normally distributed. A z-test is better suited in the case of large samples when it is more than 30. This is due to the central limit theorem that as the sample size increases, the samples are considered to be normally distributed. So for the small sample, we can go in for t-test, chi-square, and ANOVA. But then in chi-square test, we have got a condition that the expected frequencies should be less than 20%. So if in the tabulation, if you don't get that, then we are not supposed to report. So here, the testing of hypothesis, the flow chart shows the procedure. First and foremost thing, what you do is you narrate the hypothesis. You state the hypothesis, which I have described to you in the last session. You set up a suitable significance level. So normally in management, we set it as 5% level of significance. We give it we at 95%. If you generalize the data, it would be almost the same for the total population. And for very precise thing, we go in for 1% level of significance as well. Then after setting up the suitable significance level, we determine a suitable test statistics. See here, when you fix up the significance level, we generate p-values. So in the p-value, we understand how much risk we can take while you reject the null hypothesis. So you determine a suitable test statistics as well, and then you determine the critical region. We perform all the computations, and then we come to a final decision whether we accept, we reject the null hypothesis or we fail to reject the null hypothesis. So here, test of significance is the inferential statistics. So we have seen that then in the last session about the descriptive statistics, where we were talking only about the frequencies, and we were talking about the dispersion. But here in inferential statistics, we are going to get information about our immediate group of data. For example, we calculate the mean and standard deviation of the exam marks for 100 students. And this could actually provide a valuable information about this group of 100 students. But any group of data like this, which includes all data we are interested, is called the population. But the population could be small or large as long as it includes all data that we are interested into. So in inferential statistics, we could apply to find out the relationship, to find out the association, to find out correlation, and to find out variance. So in inferential statistics, it is nothing but a process of achieving. This is called sampling. Sampling strategies, actually we have discussed about it in detail. So inferential statistics arises out of the fact that sampling naturally incurs some sort of sampling error. So I was telling you already, like we have a phrase, to err is human. So we have error all the time, but we have to look into whether the error is negligible. Okay. And whether it falls within line. If it is very much erratic, then we will never take the risk of giving in false inferences for the decision makers. So the methods of inferential statistics are the estimation of parameters and testing of statistical hypotheses. So inferential statistics uses statistical methods to help you compare your sample data to other samples or to the previous research. So most research use statistical models called the generalized linear model and include students t-test, ANOVA, as I was already telling you, regression analysis, and the various other methods that result in a straight line, linear probabilities and results. So here association, testing association is through applying chi-square. So chi-square can be used to test for association between two or more variables. The chi-square test measures if there is a significant difference between the expected frequency and the observed frequency. But as I already said, there is a condition where you need to report the chi-square. So I am showing you the output of SPSS. So here you have to go on analyze, descriptive, and to the cross tabulation. So you will never find a terminology called as chi-square in the SPSS. So they name it as cross tabulation. We have got rows and columns. So it is named as cross tabulation. So you use the arrow button to move the gender variable to the rows, and reading you move it to the columns. Okay. Then after doing that, we click on the statistics button. You select chi-square and pi, and then Cramer's V, and then click to continue. Okay. Then after that, we click on the cells button. Then we just check in with the observed, then we check in with row, column, and total, because we need to know the counts, and we can actually take in the percentage as well. Okay, so finally click on OK button for the results. So nowadays it's a drop down menu, just clicking of the button, you will get the results in fraction of seconds. So from this table, the output, I am showing you the output now. From the table, the Pearson chi-square is 0.178. Okay, since the p-value is greater than 0.05. So what is it actually? The p-value is the risk of rejecting null hypothesis. So the decimals, what you get, you convert that into percentage. So when it is 0.178, it is 17 percentage. So what is that significance level we have fixed? We have fixed 5%. So only 5% risk we can take when you reject the null hypothesis. But now the result shows that we need to take 17.8% risk to reject the null hypothesis. So I will not take that risk. Okay, so what I will do, I will accept the null hypothesis. Okay, so there is no statistical significant association between gender and the reading preference of undergraduates in the university. Okay, but here the condition what I stated to you, know, like if you look at the footnote, two cells, it shows normally 50% of expected count less than 5, but the minimum expected count is 4.50. So which means that we are not supposed to report this table according to the severity. If you are very clear about it, you know, like we are not supposed to report it. Okay, that's a precondition we need to have. But just if you wanted to find out and then you report it, and you can state that in your limitations as well. Okay, so which means that some of the cells are vacant. That's what it says. Okay, so if cells are vacant, then there is no point in finding out the association because it is not been classified properly. So next comes very important and a powerful tool called as ANOVA. ANOVA is nothing but analysis of variance. We are examining if there is significant difference in the mean values of the dependent variable, which is controlled with the effect of the independent variable. So we have got only one dependent and one independent. So after taking into account the influence of the uncontrolled independent variable, essentially, ANOVA in SPSS is used as a test of means for two or more population. So ANOVA must have a dependent variable, as I've told you already, and it got an independent variable as well. And it should be categorical in nature. Okay. So in ANOVA, the categorical independent variables are called as factors. The independent variable should be put it in the factor and a particular combination of factor levels or categories, and we call it as a treatment. So if you've got two or more factors which are involved in ANOVA, then you term it as n-way ANOVA. For example, if the researcher also wants to examine the preference of total cereal by the customers who are loyal to it and those who are not, then you can use n-way ANOVA in SPSS. From the menu, we can choose that. So then you click on to analyze, go to compare means and click one-way ANOVA. The major advantage of ANOVA in SPSS is that the interactions between the independent variables could be examined properly. So the one-way ANOVA is often used to analyze data from the following studies, which states field studies, experiments, quasi-experiments as well, where you have got the control variable as well as the experimented test variable as well. So one-way ANOVA is used for two or more groups, two or more interventions, and two or more change scores as well. Okay, this is how it looks. This is how you check in. You know, like you open up the window. You will specify the variables to be used in the analysis. All the variables in the test data set appear in the list on the left side. So you have to move the variables to the right by selecting them in the list and just clicking the blue arrow button. So you can move the variable to either of two areas, so dependent list or to the factor. So the independent variables are always moved into the factor. So when you run the procedure, you add the variables print to the dependent list box and then the variable smoking to the factor box. With this is another example. So you check the box for mean plot and you click OK, then that is finished. So the output will be like this. Here you could see the significance, which is 0.000, which means that you can, you're taking only 0% risk. So you can retroactively reject the null hypothesis. So the ANOVA alone does not tell us specifically which means were different from one another, but it just tells us whether there is a significant difference or not. Then comes independent sample t-test. So I told you, for small samples, which is less than 30, we can use t-test. So the independent sample t-test window opens where you will specify the variables to be used in the analysis. So all the variables in the data set appear in the list on the left side. So you move the variable to the right side by selecting them in the test variables or the grouping variable, and then you go in for clicking the button. Okay. When you click the button, then everything is finished. Okay, so you go compare the means, you group the variable, you test the variable, define groups, use specified values, then click OK, you get the results. Okay. Thank you guys. See you in next class.